ओम हम धन्वंतरे नमः। Topic: Leprosy and psoriasis histopathology. So here, histopathological study of leprosy and psoriasis is discussed. Leprosy mainly two types: lepromatous leprosy and tuberculoid leprosy. This is a slide of lepromatous leprosy. Lepromatous leprosy (LL) histopathology is characterized by a dense, diffuse infiltrate of foamy macrophages, that is histiocytes, spanning the dermis. A hallmark feature is the clear Grenz zone, a spared band of normal collagen between the epidermis and the infiltrate. Special stains, VAD fight, reveal abundant Microbacterium leprae grouped into dense balls called globi. For visual examples, detailed microscopic slides, and diagnostic references, you can examine the web pathology lepromatous leprosy image, which illustrates the classic foamy histiocytes and Grenz zone. For a broader range of Hansen's disease histopathology, including comparisons to tuberculoid and borderline subtypes, the DermNet leprosy pathology resource offers comprehensive microscopy breakdowns. Tuberculoid leprosy. Tuberculoid leprosy histopathology reveals well-defined sarcoidal granulomas in the dermis composed of epithelioid histiocytes and Langhans gene cells surrounded by a dense cup of lymphocytes. A key diagnostic feature is the invasion and destruction of cutaneous nerves by this granulomatous process. Additionally, the bacilli are typically scarce or absent. For visual examples of these microscopic features, you can browse slides on web pathology. Diagnostic criteria include epithelial granulomas, compact collections of activated macrophages that push directly up against the epidermis. There is no Grenz zone. Giant cells. Multinucleated Langhans giant cells are frequently present within the granulomas. Nerve destruction. Granulomas infiltrate and destroy dermal nerve bundles, causing the characteristic loss of sensation in skin lesions. Scanty bacilli. Unlike lepromatous leprosy, acid-fast bacilli stains, example Wade 5, are typically negative or show only rare fragmented bacteria. Tuberculoid. Next, gutted psoriasis. The histopathological study of gutted psoriasis. The histopathology of gutted psoriasis is characterized by mild acanthosis, thickening of the epidermis, uniform elongation of the rete ridges, and prominent dilated papillary dermal blood vessels. It also features parakeratosis, retention of nuclei in the stratum corneum, containing collections of neutrophils known as Munro's microabscesses. Second type is gutted psoriasis. This is gutted psoriasis. Next is plague psoriasis. Plague psoriasis. Histopathology slide look like this. Plague psoriasis histopathology is classically characterized by regular epidermal hyperplasia with elongation of the rete ridges, often described as a test tube in a rack appearance, parakeratosis, and thinning of the suprapapillary plates. Key microscopic diagnostic features. Medical professionals evaluate a hematoxylin and eosin stained skin biopsy for several hallmark features. Acanthosis and rete ridges. Regular elongation and thickening of the epidermal rete ridges that downwardly project into the dermis at uniform lengths. Parakeratosis. Retention of cell nuclei within the stratum corneum, outermost skin layer, typically arranged in focused molds. Hypogranulosis, a distinct thinning or complete loss of granular layer beneath areas showing parakeratosis. Munro microabscesses, microscopic collections of neutrophils trapped specifically within the parakeratotic stratum corneum. Coggous pustules, spongiform pustules formed by neutrophils migrating into the upper spinous layer of the epidermis. Dermal changes. Dilated tortuous capillaries extending high up into the dermal papillae, which are accompanied by a perivascular lymphocytic infiltrate. The lake psoriasis. So these are taken from the internet, AI and other sites. So thank you for patient hearing.